Major histocompatibility complex class 1 molecule play an important role in the immune system, especially in the adaptive immune response. These molecules are important for the presentation of intracellular antigens to cytotoxic T cells, enabling the immune system to recognize and eliminate the infected cells or the abnormal cells. Understanding the structure, function, and regulation of class 1 MHC molecules is essential for understanding the intricacies of immune surveillance and human defense mechanisms. At a fundamental level, MHC class 1 molecules are glycoproteins expressed on the surface of most nucleated cells in our body. They consist of a polymorphic heavy chain encoded by genes within the MHC locus and a non-polymorphic beta-2 microglobulin beta-2M chain. The heavy chain comprised of three extracellular domains, alpha-1, alpha-2, and alpha-3. Alpha-1 and alpha-2 domains form a peptide binding groove where antigenic peptides, typically 8 to 10 amino acids in length, are presented to T cells. This peptide representation or presentation is essential for the activation of cytotoxic T cells, which are important in eliminating infected or malignant cells. Alright, let's understand this process. So the step 1 here is the peptide loading. Peptide loading onto the MHC class 1 molecule occurs inside the endoplasmic reticulum organelle. So endoplasmic reticulum is very important here. It is facilitated by multi-subunit peptide loading complex, and this complex is involved in loading the peptide onto the MHC class 1 molecule. This process involves the trimming and selection of suitable peptides from a pool of potential antigens generated within the cell. Peptides that binds tightly, specific peptides, they have the ability to bind to the MHC class 1 tightly. They are selected, they are retained, and others, they are released. The peptide-loaded MHC class 1 molecule complexes are then trafficked to the cell surface where they await inspection by cytotoxic T cells. The diversity of peptides presented by MHC class 1 molecule is influenced by several factors, including the peptide binding specificity of MHC molecule itself, the abundance and turnover rate of extracellular proteins, as well as the activity of proteasomes with other proteolytic enzymes involved in antigen processing. Additionally, the expression level of various proteins like trypsin and chaperones like Calreticulin plays an important role in regulating peptide loading process and MHC class 1 assembly. Alright everyone, that was all about MHC class 1 molecule. We have discussed the structure, we have discussed the function. I'll meet you in the next one where we will discuss interesting topics like this. If you have liked the video, then please do hit the like button, and if you're new to the channel, then please do subscribe to Basic Science series.